At any given moment, a living cell is bombarded by a chaotic barrage of chemical signals. Hormones, neurotransmitters, and autocoids wash over the exterior membrane, carrying instructions from the rest of the body. To translate this extracellular chaos into an organized intracellular response, the cell relies on a superfamily of membrane proteins known as G-Protein Coupled Receptors, or GPCRs. While these receptors bind wildly different ligands on the outside, they share a conserved structural organization to process that information on the inside. The core architecture consists of 7 hydrophobic alpha helical segments that weave directly through the lipid bilayer. This structure creates an amino terminus on the extracellular face to catch the incoming ligand, and a carboxy terminus on the cytosolic side to pass the message along. That message is handed off to the system's engine, a heterotrimeric G-protein complex floating freely along the inner membrane surface. By coupling this 7 transmembrane architecture to a select few intracellular pathways, GPCRs act as biological signal amplifiers. They take a transient exterior event, a single molecule brushing against a cell, and convert it into widespread interior physiological changes. In its baseline state, the G-protein is a heterotrimer with GDP bound to the alpha subunit. An agonist binding the exterior receptor locks its cytosolic loops onto the G-protein. This triggers nucleotide exchange, replacing GDP with GTP, causing the alpha subunit to immediately break away. The structure is now split into an independent alpha GTP complex and a free beta-gamma dimer. Both are now fully active, migrating along the membrane to interact with different intracellular effectors. But biological systems require constraints. A cellular signal cannot run indefinitely. To prevent runaway activation, the alpha subunit possesses intrinsic GTPase activity. Over a period of several seconds, the alpha subunit slowly hydrolyzes its bound GTP, converting it back into GDP. This chemical reduction forces the alpha subunit to deactivate and reassociate with the beta-gamma dimer. This intrinsic GTPase activity functions as a strict molecular timer. It ensures the amplification cascade lasts long enough to be effective, then shuts off automatically. This diagram illustrates a myocardial cell's action-effect sequence. On the left, adrenaline binds a beta-adrenergic receptor, which couples to a stimulatory G-protein, GS. The activated GS alpha subunit then targets the membrane enzyme adenylyl cyclase. Adenylyl cyclase converts available intracellular ATP into the second messenger cyclic AMP, or CAMP. The primary function of this newly generated CAMP is to find and activate protein kinase A. Active protein kinase A modifies membrane calcium channels to increase influx and alters troponin and phospholambin. Physiologically, this synchronized targeting leads directly to increased cardiac contractility and faster relaxation. Other receptors achieve the opposite effect by coupling to GI, or inhibitory proteins. The muscarinic M2 receptor uses a GI alpha subunit to inhibit adenylyl cyclase, immediately suppressing CAMP production. Triggering a cascading kinase network allows a single GS alpha activation to force widespread protein phosphorylation, massively amplifying the heart's contractile force. This schematic details the phospholipase C pathway in a smooth muscle cell reacting to histamine via the H1 receptor. Upon activation, the GQ alpha subunit targets phospholipase C beta. This enzyme hydrolyzes PIP2, splitting the specialized lipid into 2 distinct second messengers, IP3 and DAG. The water-soluble IP3 diffuses deep into the cytosol, triggering a release of stored calcium from the endoplasmic reticulum. This sudden flood of cytosolic calcium binds to calmodulin, which in turn activates myosin light chain kinase, driving the physical contraction of the smooth muscle. Meanwhile, the second messenger, DAG, is lipophilic. It remains embedded in the membrane and works in conjunction with that newly released calcium to activate protein kinase C. Cleaving a single phospholipid creates a dual-pronged effector wave, mobilizing deep calcium stores for contraction while activating peripheral kinases to alter broader cellular functions. GPCRs do not always rely on complex secondary messenger cascades like CAMP or IP3 to alter cellular function. Returning to the myocardial diagram, we examine the muscarinic M2 receptor. When acetylcholine activates the inhibitory GI protein, the green beta-gamma dimer breaks away. Instead of seeking an enzyme, it directly opens nearby potassium channels. Opening these specific potassium channels causes a rapid efflux of potassium ions. This hyperpolarizes the myocardial cell, depressing electrical impulse generation without a single secondary messenger being synthesized. The G-protein is a dual-action transducer. The alpha subunit manages dense enzymatic cascades, while the beta-gamma dimer secures immediate electrical control over the cell membrane. Stepping back from these microscopic enzymatic pathways, we can observe the macroscopic reality of the cell membrane, where thousands of different chemical signals arrive simultaneously. GPCR signaling represents a biological economy. An immense variety of external inputs are entirely managed by one conserved 7-transmembrane architecture. By forcing all these varied receptors to share a strictly limited pool of transducer proteins, specifically GS, GI, GO, and GQ, the cell creates a signaling bottleneck by design. Because so many distinct receptors utilize the same few internal pathways, the cell is able to physically integrate the data. It calculates the sum total of diverse and sometimes opposing signal inputs simultaneously. GPCRs function as the cell's primary processing units, converting the chaos of the extracellular environment into a precisely timed, integrated biochemical reality.